Get Private Johnson some more plasma, Stat.
Yes, nurse. Right away. He's not improving. We're nearly out of penicillin.
I know.
We just have to hope the convoy makes it. Keep that leg elevated and watch for infection.
Nurse, prepare the next dosage immediately.
Right away, Doctor.
The pressure is fluctuating beyond acceptable limits, Inspector. We can't stabilize the batch.
I don't care about the limits, Doctor. We need the penicillin now.
This penicillin will have you back on your feet in no time.
I appreciate it, Doc. When can I get back to the front?
The readings are steady right here, Mr. B.
Adjusting the valve now. Inspector, the yield projections are on schedule.
See that they stay that way, Doctor. The front needs this.
January 1943. A makeshift hospital near the docks of New York hums with quiet urgency. Outside, snow falls in pale sheets, settling on stacked crates of military supplies, while inside the air smells sharply of antiseptic and the faint iron tang of blood. Nurses move quickly between rows of cots, their shoes squeaking against the linoleum. Soldiers, fresh from battlefields in North Africa and the Pacific, lie pale and shivering, bandages soaked, wounds infected. A young private, his leg wrapped in crude gauze, winces as a nurse tends to him. "If only we had more. Something," he mutters, eyes darting to the empty shelves where antibiotics should be. Doctors exchange concerned glances. Infections that should be survivable are creeping into mortal threats. Each hour, the absence of penicillin, the miracle drug first discovered 15 years prior, feels heavier. The clock ticks. Outside, ships thunder along icy harbors, carrying men and machines to distant, brutal fronts. Inside, the question presses: how could America arm its soldiers with life-saving medicine it barely possesses? The race against time is about to begin, and one company holds a secret that could change the course of the war. By early 1943, the world was engulfed in a total war unlike any before. On European soil, the clash between Axis and Allied forces had reached a critical stage. American soldiers were engaged in North Africa, pushing against entrenched German and Italian positions, while the Pacific Theater demanded relentless naval and jungle campaigns against a determined Japanese enemy. Across both continents, battlefield wounds were routine. Infections, often as lethal as bullets, were a constant shadow over every operation. Medical care had advanced in the decades prior, yet remained painfully inadequate against the virulent infections that followed modern warfare. Gangrene, pneumonia, and septicemia claimed thousands of lives in military hospitals. Conventional treatments, sulfa drugs, iodine, and crude antiseptics, offered limited efficacy. Each day, doctors faced the grim choice of amputating limbs or watching soldiers succumb to infections that, in a laboratory setting, were preventable. The discovery of penicillin in 1928 by Alexander Fleming had promised a new era of medicine. In its early experimental use, the mold-derived antibiotic showed near-miraculous effects, neutralizing bacteria that had once been fatal. Yet in 1943, the United States had only scratched the surface of production. Fleming's discovery remained largely a laboratory marvel, insufficiently tested and too fragile for mass application. The military's demand was growing exponentially, outpacing the meager supplies from laboratories scattered across the country. American pharmaceutical companies were suddenly thrust into a critical mission: produce life-saving penicillin at an industrial scale and do it fast. Among them, Merck Co, one of the nation's oldest chemical and pharmaceutical firms, emerged as a key player. The challenge was not merely scientific, it was logistical and moral. Scientists faced the daunting task of scaling a delicate fermentation process, a method that had only worked in small laboratory flasks, into vats capable of generating hundreds of liters of pure penicillin. Each stage, controlling temperature, pH, oxygen, and contamination, was precarious. A single misstep could destroy weeks of work, setting the production timeline back by months. Government agencies were acutely aware of the stakes. The Office of Scientific Research and Development, OSRD, which coordinated wartime research, liaised with Merck and other pharmaceutical companies, offering guidance and stringent oversight. Contracts were issued, incentives provided, yet the pressure was relentless. Military planners emphasized the urgency. D-Day was on the horizon. North African campaigns were ongoing, and casualties continued to mount. Every lost soldier due to infection was a potential loss of strategic advantage. Across the Atlantic, European hospitals struggled under similar pressures. The British and Canadians had begun small-scale penicillin production with experimental strains, but output was minimal. The American approach needed to be transformative. To meet the scale required for Allied operations, chemists and engineers had to innovate. They experimented with different strains of Penicillium mold, developed new deep-tank fermentation techniques, and devised methods to purify the fragile compound without destroying its efficacy. It was an unprecedented fusion of science and industrial engineering, a race where victory meant lives saved and defeat meant preventable deaths. At Merck's laboratories, nights stretched into days. Researchers huddled over fermenters, notebooks filled with meticulous observations, eyes scanning for contamination that could compromise entire batches. Engineers modified vats, introduced air circulation systems, and coordinated with chemists to stabilize penicillin for transport. Simultaneously, government inspectors monitored each step, ensuring compliance with evolving standards. Delays were costly, not only financially, but morally, as every day without adequate penicillin translated to lives lost on distant battlefields. Meanwhile, soldiers and medics on the front lines were unaware of the industrial and scientific struggle unfolding thousands of miles away. In hospitals from Cairo to New Guinea, they encountered the stark reality of infection. Nurses, exhausted from relentless shifts, treated gangrenous wounds with what little they had: sulfa powder, iodine, crude surgical techniques. Even small doses of experimental penicillin, often smuggled in from laboratory trials, were treasured and rationed, administered to those in the most desperate condition. Commanders took note. Reports showed that infection rates were crippling unit effectiveness. The urgent need for large-scale production was no longer an academic or corporate challenge. It was a tactical imperative. This period also highlighted the broader transformation of American industry during wartime. Factories once producing consumer chemicals or pharmaceuticals pivoted almost overnight to support the war effort. Coordination between government, industry, and scientists became a model of centralized efficiency. Each department of Merck, from microbiology to chemical engineering to logistics, had to synchronize flawlessly. Even minor misalignments, delayed raw materials, miscalculated fermentation schedules, or shipping errors could ripple across the system, undermining the goal of mass supply. The company's leadership recognized that success required both scientific ingenuity and unwavering discipline. By mid-1943, these efforts began to bear fruit. Pilot production runs of penicillin demonstrated that, with careful control, vats could yield sufficient quantities to meet military demands. The breakthrough was incremental but tangible. Reports from medical officers hinted at remarkable recoveries, even among soldiers whose wounds had seemed hopeless. The stage was set for an industrial and medical revolution. The combination of human ingenuity, scientific perseverance, and logistical precision promised to bring the miracle of penicillin to thousands of soldiers and, eventually, civilians worldwide. Yet, the story was far from over. Scaling production from pilot runs to mass supply was fraught with challenges. decisions about allocation, who would receive the limited doses first, posed moral dilemmas. Strategic choices had to be made. Frontline soldiers or field hospitals. North Africa or the Pacific Theater. Every vial represented more than medicine. It represented a potential pivot point in the war itself. The weight of responsibility rested on scientists, engineers, and military planners alike. In this crucible of necessity, human lives intersected with science, technology, and leadership. The scarcity of penicillin in 1943 was not merely a medical problem. It was a national crisis, a test of America's capacity to innovate under pressure. Each experiment, each fermenter adjustment, each shipment prepared for transport was a step toward bridging the gap between human need and technological possibility. The tension between what was available and what was required would define the coming months and set the stage for a turning point that would reshape wartime medicine forever. The war raged on outside laboratory walls, but inside Merck's facilities, a quiet revolution in medicine was taking shape. The race to produce penicillin at scale was underway, with stakes higher than any before. Lives, limbs, and the very momentum of Allied forces depended on its success. The human, technological, and strategic angles of this story would soon converge, culminating in breakthroughs that would change the trajectory of both the war and medical history itself. The story of penicillin in 1943 was not just about laboratories or chemical formulas. It was fundamentally about people. On the front lines, the human stakes were stark and immediate. Soldiers wounded by bullets or shrapnel often faced infections more lethal than their injuries. Nurses worked long, exhausting hours in dimly lit wards, tending to men whose cries of pain echoed through hallways lined with cots. Each patient represented a ticking clock. Untreated infections could claim lives in days. When even small amounts of penicillin arrived from experimental batches, the relief was palpable. Soldiers and nurses alike described a sense of almost miraculous hope, a tangible difference in the grim calculus of survival. Letters home carried fragments of this miracle. Private soldiers wrote of wounds that would have necessitated amputations now healing, of fevers breaking, of lives spared. These personal stories underscored the profound human dimension of the penicillin crisis. It was not merely chemical innovation, but a lifeline for thousands. Behind the scenes, the technological challenges were equally daunting. Penicillin, delicate and volatile, resisted easy mass production. Small-scale lab experiments had worked, but translating these methods into industrial volumes required unprecedented ingenuity. Scientists experimented with hundreds of strains of Penicillium mold, searching for those capable of higher yields. Fermentation, a process involving precise control of temperature, pH, oxygen, and nutrient concentration, demanded constant monitoring. Even minor contamination could destroy weeks of effort. Engineers worked alongside chemists to design vats and pipelines capable of supporting continuous, large-scale fermentation without compromising the compound's efficacy. The process became a delicate choreography, blending chemistry, microbiology, and mechanical engineering in ways never attempted before. Each technical innovation, stabilization methods, filtration techniques, air circulation improvements, was a small victory against the invisible enemy of production inefficiency. Leadership at both corporate and government levels played a pivotal role in driving these efforts forward. Merck's executives had to balance scientific caution with wartime urgency. They coordinated teams across departments, ensuring that microbiologists, engineers, and logistics managers worked in concert. Simultaneously, government officials applied both pressure and support, offering funding, oversight, and high-level guidance through the OSRD. Decisions were urgent and sometimes fraught, whether to divert raw materials from civilian production to military supply, how to prioritize shipments, and went to risk sending partially tested batches to the front lines. The weight of responsibility rested heavily on these leaders. Their decisions shaped not only production schedules, but potentially the lives of thousands of soldiers, turning corporate management into a matter of national and moral consequence. The military, for its part, viewed penicillin as a tactical asset. Reports from field hospitals highlighted that even small doses drastically improved survival rates, shortened recovery times, and allowed soldiers to return to combat more quickly. Commanders soon realized that the availability of penicillin could influence operational planning. An advance into enemy territory was no longer only a question of firepower or logistics. It now involved calculations about medical support and infection control. The impact on troop morale was equally significant. Soldiers knew that a serious wound did not automatically mean death or permanent disability, a psychological reassurance that reshaped the way battles were fought and endured. From the enemy perspective, penicillin was an invisible force shaping the battlefield. Axis planners could not account for it, yet it indirectly altered the strategic landscape. Units fortified by better medical care recovered faster, maintained higher fighting capacity, and sustained fewer losses from disease and infection. The Allies' increasing access to this miracle drug meant that attritional calculations for battles had changed. What might once have been a crippling casualty count was now manageable, shifting momentum in subtle but decisive ways. Moral dilemmas, too, emerged alongside technological triumphs. With penicillin in limited supply, decisions had to be made about allocation. Which soldiers or units would receive doses first? Should priority go to officers, frontline troops, or the severely injured? These choices were never abstract. They were daily realities in hospitals and labs alike. Diaries and letters from nurses and doctors reveal the weight of these decisions. Each vial distributed was a life saved, but each vial withheld potentially meant a life lost. The human and ethical dimensions intertwined inseparably with industrial and technological challenges, creating a tension that underpinned every breakthrough. The technological, human, leadership, military, enemy, and moral angles converged in a high-stakes environment that demanded rapid, coordinated action. Each aspect reinforced the others. Scientific innovation enabled greater human survival. Leadership decisions dictated effective distribution. Tactical planning depended on medical reliability, and moral judgment guided the fair allocation of a scarce resource. The unfolding story of penicillin production was therefore a multi-layered drama, blending human courage, scientific brilliance, and industrial precision. As summer 1943 approached, these angles were not merely theoretical. They began manifesting tangible results. Pilot batches of penicillin emerged from Merck's vats, and initial shipments reached field hospitals. Reports from North Africa and the Pacific noted fewer amputations, quicker recoveries, and dramatically improved survival rates. For soldiers and medics, the presence of penicillin was not an abstraction, but a daily lifeline. The human impact was visible. Young men who would have faced death or permanent injury now returned to duty, altering the course of campaigns. Simultaneously, engineers and scientists refined production methods, scaling output to meet growing military demand. Leadership coordinated shipment and allocation strategies, ensuring the medicine reached the most critical fronts first. This convergence of angles set the stage for a decisive turning point. The delicate balance between human need, technological capacity, and strategic urgency would soon tip toward success or failure. The race to deliver mass penicillin production was no longer an experiment. It was a pivotal moment in wartime medicine, where lives hung in the balance, and the outcome depended on precise coordination across multiple fronts. The breakthroughs in laboratories and factories promised to transform not just hospitals, but the battlefield itself, creating a new paradigm in medical support for war. By late 1943, it became clear that the combined efforts of human courage, technological ingenuity, and decisive leadership were converging toward a moment of unprecedented impact. The production challenges that had seemed insurmountable were yielding to disciplined scientific and industrial effort. The first large-scale shipments of penicillin were poised to reach frontline hospitals in meaningful quantities. Soldiers would experience the tangible benefits of these efforts within weeks, marking the culmination of months of painstaking work. Every angle, the human, technological, leadership, tactical, enemy, and moral, interlocked, creating a story not only of science and industry, but of survival, strategy, and ethical decision-making. This intricate web of human and technological effort naturally led to a climax. The moment when penicillin transitioned from scarce experimental batches to a mass-produced lifeline capable of altering the course of the war. The tension, uncertainty, and stakes of this period would soon give way to a transformative turning point, a moment where months of labor and ingenuity would directly save lives on a scale never before achieved in military medicine. By mid-1943, the tension surrounding penicillin production had reached a fever pitch. In Merck's laboratories in Rahway, New Jersey, scientists and engineers worked in a coordinated frenzy. The vats, once experimental glass flasks, had been scaled up to steel fermenters capable of holding hundreds of liters. Each batch required constant vigilance. Temperature, pH, and oxygen levels had to be meticulously monitored, while contamination remained a constant enemy. A single slip could ruin an entire run, wasting weeks of effort and precious mold cultures. Dr. Jasper Kane, a lead chemist at Merck, oversaw one of the critical fermentation lines. He moved between tanks with careful precision, checking readings, making adjustments, and instructing assistants on delicate procedures. Keep the oxygenation steady. Any fluctuation could collapse the yield, he murmured, his voice tense but measured. Outside the lab, government inspectors from the OSRD monitored every step. Their presence added pressure, but also validated the enormity of what was at stake. The lives of thousands of soldiers on distant battlefields depended on success here. Meanwhile, engineers had implemented innovative systems to stabilize the fragile penicillin. Specially designed air filtration units minimized contamination, and novel filtration techniques allowed the compound to be extracted without losing potency. It was a marriage of biology and industrial engineering, a technical symphony unfolding under the relentless pressure of war. Pilot runs had shown promise, but scaling to mass production remained uncertain. Could the processes hold for sustained daily output? And could these batches survive shipment across oceans to field hospitals? The first breakthrough came quietly in late summer 1943. A series of fermenters produced a remarkably high yield, and analysis confirmed that the penicillin maintained full potency. The laboratory erupted in restrained celebration. Scientists knew this was the turning point they had been striving for. Within days, additional vats matched the yield, demonstrating that the production process was now reliable enough for larger-scale distribution. Government officials immediately approved the first major shipment to military hospitals overseas. The logistical operation that followed was unprecedented. Vials of penicillin, carefully packaged to preserve stability, were transported to ports, loaded onto ships bound for North Africa, the Pacific, and eventually the European front. Each shipment was accompanied by detailed instructions for storage and administration. Improper handling could render the precious medicine useless. For soldiers in the field, this supply represented a literal lifeline. Wounded men who might have faced amputations or death from infection now had a chance at survival. Field hospitals began receiving their first substantial shipments by late 1943. In North Africa, medics at a hospital near Algiers were among the first to witness the impact. Corporal Richard Hayes, recovering from shrapnel wound in his leg, described the change. The doctors injected me with this new medicine. Within days, the fever broke, the swelling subsided. I didn't lose my leg. Similar accounts emerged across campaigns in the Pacific, where Japanese artillery and disease posed constant threats. The transformation was immediate and visible. Where infection had once devastated units, soldiers now recovered quickly, reducing both mortality and long-term disability. The military quickly recognized penicillin as a strategic asset. Commanders adjusted medical protocols, ensuring that the most critical cases received treatment first. The drug's availability allowed for more aggressive operational planning. Offensives could proceed with reduced risk of infection decimating units. Intelligence reports noted that unit effectiveness and morale were improving in hospitals that received regular shipments. The Allies' ability to sustain combat operations under adverse conditions had increased significantly. Penicillin had shifted from experimental curiosity to essential weapon of war. The production success also reshaped industrial priorities. Merck, alongside Pfizer, Squibb, and other pharmaceutical firms, rapidly expanded fermentation capacity. Additional plants came online, and novel extraction and stabilization methods enabled shipment in larger quantities. Scientific teams worked around the clock, supported by engineers who maintained the complex equipment. The combined effort of chemists, engineers, logisticians, and government officials created a production chain unlike any the world had seen before. Each day, thousands of additional doses left the factories, reaching front lines where they would save countless lives. Amid these triumphs, the human element remained central. Nurses in field hospitals began to see dramatic reductions in mortality from infections. Medics reported that wounds which previously required amputations were now healing fully. Soldiers, many of whom had lost friends to infections that could have been treated, found renewed hope. Letters home recounted the miracle of recovery. Young men who might have been permanently maimed now returned to duty, carrying with them stories of a medicine that seemed almost magical. This human response underscored the moral and emotional weight of the scientific achievement. Yet challenges persisted. Shipping across oceans was perilous. Submarines, storms, and logistical delays threatened to interrupt supply lines. To mitigate risk, convoys were prioritized and protective packaging innovations were introduced to preserve the penicillin's potency. Even small mishaps could have immediate consequences on survival rates at field hospitals. The combination of scientific precision, industrial efficiency, and logistical coordination was tested continuously, reinforcing the precariousness of the operation. By early 1944, the full scale of the achievement became apparent. Merck and its partners were producing enough penicillin to meet the majority of military requirements. Mortality from infected wounds plummeted. Battlefield reports indicated reductions as high as 50 to 70% in some theaters. Doctors and medics lauded the medicine as transformative. The psychological impact on soldiers was equally profound. Fear of infection, once an omnipresent anxiety, was replaced by cautious confidence. Penicillin had not only saved lives, but strengthened the fighting capacity of entire divisions. The success also had far-reaching implications beyond immediate military utility. Pharmaceutical manufacturing had been tested under unprecedented conditions, resulting in innovations in fermentation, purification, and quality control. These techniques would later fuel the post-war expansion of antibiotics for civilian use, democratizing access to a medicine that had previously been experimental and scarce. But in 1943 to 44, the focus remained intensely narrow, saving soldiers and ensuring that Allied operations could proceed unimpeded. The climax of this story was therefore both human and industrial, tactical and technological. In laboratories, vats of penicillin represented months of labor, precision, and ingenuity. In hospitals, Vials of the drug represented lives saved, wounds healed, and morale restored. On the front lines, it transformed operational planning, reducing the invisible casualties caused by infection. The convergence of scientific innovation, industrial mobilization, leadership decisions, and human resilience culminated in a pivotal moment. The successful mass production and deployment of penicillin at a scale previously thought impossible. As the first waves of penicillin reached frontline hospitals, the race against infection and mortality was turning decisively in favor of the Allies. The months of uncertainty, experimentation, and labor had produced a tangible, life-saving outcome. The technological triumph had immediate human consequences. Soldiers who might have been lost to infection were now returning to duty, altering the dynamics of campaigns in North Africa, the Pacific, and soon, Europe. This decisive breakthrough set the stage for the aftermath of mass penicillin deployment. Strategic advantages were solidified, mortality rates dropped, and the moral and psychological landscape of the war shifted. Yet the story did not end with production. The consequences of this medical revolution, the transformed battlefield, the reshaped pharmaceutical industry, and the ethical decisions surrounding allocation were only beginning to unfold. The triumph of penicillin in 1943 to 44 would reverberate through the war and beyond, leaving a legacy that bridged science, human resilience, and the relentless demands of total war. The arrival of mass-produced penicillin on the front lines marked a turning point not only in medicine, but in the very calculus of war. Hospitals, once overwhelmed with infections that claimed limbs and lives indiscriminately, began to see a transformation. Mortality rates among wounded soldiers dropped dramatically. Reports from field hospitals in North Africa indicated that fatalities from septic wounds fell by nearly half within months of the first shipments. By the spring of 1944, similar patterns were emerging in the Pacific theater, where Japanese artillery and tropical diseases had previously created near-constant medical crises. Soldiers who would have faced amputation or death now recovered fully, often returning to combat within weeks. The human cost of infection was being rewritten. Nurses and medics on the ground noted not just a reduction in deaths, but also a shift in morale. The psychological burden of witnessing patients succumb to infections, once a daily reality, eased measurably. Corporal Richard Hayes, who had received penicillin for a shrapnel wound in his leg, described the effect on both patients and staff. It was like a veil lifted. We could treat wounds with hope, not dread. Men began to smile again, even in the midst of war. Diaries and letters from nurses echoed this sentiment, recounting the relief and almost reverent gratitude for the drug that had arrived just in time to save countless lives. Strategically, the widespread availability of penicillin altered military planning and operations. Commanders could now assume that infection would no longer incapacitate units as severely as before. Offensive operations, which had previously been tempered by the risk of medical attrition, became more feasible. Field hospitals could handle casualties with greater confidence, and wounded soldiers could return to active duty sooner. The Allies' overall combat effectiveness increased, a subtle yet significant shift that contributed to subsequent successes in Sicily, Italy, and eventually the Normandy landings. The invisible weapon of mass-produced penicillin had reshaped the battlefield, providing an advantage no enemy force could counter directly. The quantitative impact was staggering. By mid-1944, Merck and its partners, including Pfizer, Squibb, and several smaller companies, were producing hundreds of millions of units of penicillin annually. The FDA and OSRD closely monitored distribution to ensure that the most critical areas received priority. Tens of thousands of soldiers had already benefited from early doses, and projections indicated that the vast majority of frontline personnel would soon have access. In operational reports, Medical officers noted that wound infections that previously incapacitated entire companies were now manageable. Field hospitals could perform more surgeries, save more limbs, and reduce recovery times. The statistical shift was undeniable. Penicillin was not a marginal improvement. It was a revolution in military medicine. The success of mass production also reinforced the relationship between industry and government. The collaboration between Merck, other pharmaceutical firms, and federal agencies became a model for large-scale wartime coordination. Companies adapted factories and staff to maintain relentless output, while government agencies facilitated raw material supply, transport, and regulatory oversight. This unprecedented synchronization demonstrated that industrial capacity, when combined with scientific innovation and strategic urgency, could overcome even the most formidable challenges. The wartime push for penicillin production set new standards for collaboration, efficiency, and problem-solving under extreme pressure. Ethical and moral considerations persisted even as production ramped up. Supply, though vastly improved, was still finite, requiring prioritization. Military hospitals had to decide which patients would receive doses first. The ethical calculus often favored the most severely wounded or those whose recovery would have the greatest operational impact. While the distribution was largely successful, nurses and doctors recounted difficult decisions, moments when the scarcity of medicine forced them to choose between lives. These dilemmas underscored that penicillin was not merely a scientific triumph, but also a moral one. The responsibility of allocating life-saving medicine weighed heavily on those administering it. Beyond the immediate war front, the impact of penicillin reshaped the medical and pharmaceutical landscape. The industrial fermentation methods developed to scale production proved adaptable for post-war civilian use. Hospitals and clinics began receiving larger supplies of penicillin, dramatically reducing deaths from bacterial infections across the population. The success of wartime production accelerated research into new antibiotics, creating a legacy of innovation that would define medicine for decades. The triumphs achieved under the urgent pressures of war demonstrated that necessity could drive scientific and industrial breakthroughs with long-term societal benefits. Field reports from the European and Pacific theaters highlighted these outcomes vividly. In a hospital near Naples, Italian campaigns benefited directly from newly supplied penicillin. Doctors treated wounds that had previously resulted in severe disability, and patients were discharged sooner, restoring morale and operational efficiency. In the Pacific, where tropical diseases had compounded battlefield injuries, penicillin proved equally transformative. Mortality rates for infected wounds, gangrene, and pneumonia fell sharply. Soldiers who had faced near-certain death now had a real chance to survive. Each vial delivered across oceans, each dose administered with care, represented not only a triumph of science and logistics, but a triumph of human perseverance in the face of adversity. Statistical assessments underscored the broader significance. The mortality rate from infected wounds, which had hovered around 30 to 40% in some theaters before penicillin, dropped to under 10% within months of widespread distribution. Recovery times shortened, amputations declined dramatically, and overall troop readiness improved measurably. Commanders noted that the presence of penicillin allowed for more aggressive operations and reduced the psychological toll of combat. In effect, the drug became a silent ally on the battlefield, saving lives and influencing the course of campaigns in ways that artillery and armor alone could not achieve. The industrial triumph was equally remarkable. Merck's Rahway plant, alongside its partners, demonstrated that large-scale chemical production could be achieved without sacrificing quality. The innovations in deep tank fermentation, purification techniques, and quality control were unprecedented. Each production line represented a fusion of chemistry, engineering, and management, all driven by the urgency of wartime necessity. The lessons learned here would inform peacetime pharmaceutical manufacturing, creating a lasting legacy that extended far beyond the immediate demands of the war. By late 1944, the success of penicillin production was no longer an isolated achievement. It had become a central pillar of Allied medical and operational strategy. Soldiers, medics, and commanders had all adapted to the presence of this life-saving medicine, and its effects were felt across multiple theaters of war. Reports from the front lines emphasized not just the physical healing it provided, but the psychological reassurance it offered. Soldiers fought with renewed confidence, knowing that a severe wound no longer guaranteed death or permanent disability. The aftermath of penicillin's mass production illustrates the interconnectedness of science, industry, leadership, and human resilience. What began as a laboratory challenge evolved into a full-scale industrial operation, saving lives and influencing military strategy. The ethical decisions surrounding distribution, the technical innovations in production, and the human stories of recovery all converged to create a profound, multifaceted impact. The success of 1943 to 44 established penicillin as a transformative force in modern warfare, demonstrating that medicine could be as decisive as guns, tanks, and planes in determining the course of history. As the war continued, penicillin's presence on the battlefield reshaped not only outcomes, but expectations. Soldiers, medics, and leaders recognized that scientific innovation could directly influence survival and strategy. Hospitals became centers of hope rather than despair. Factories became engines of life-saving production. And in the interplay of these forces, the Allies found a new advantage, one that would ripple across campaigns and continue to shape military medicine long after the guns fell silent. The transformation wrought by penicillin naturally leads to reflection. Beyond statistics, battles, and strategic calculations, the story of this medicine encapsulates the resilience of humanity, the power of innovation under pressure, and the enduring consequences of decisions made in moments of extreme necessity. From laboratory vats to frontline hospital beds, the human, technological, and moral dimensions converge to create a legacy that continues to echo through history. The story of penicillin in 1943 to 44 is a testament to human ingenuity under pressure. In the midst of a global conflict where bullets and bombs determined life and death, it was a delicate mold in a laboratory that transformed the battlefield. The scarcity of penicillin forced scientists, engineers, military planners, and medical personnel into a relentless race against time. Every fermenter, every shipment, every decision about allocation carried weight far beyond a laboratory or the factory floor. Lives hung in balance, and the stakes could not have been higher. Yet the triumph was not purely technical. It was profoundly human. Soldiers whose wounds would have been fatal recovered. Nurses, once exhausted and despairing, could treat patients with hope rather than fear. Commanders could plan offensives knowing that infection was no longer an insurmountable enemy. In each vial of penicillin lay the convergence of courage, precision, and determination. It was a reminder that even in the most destructive of human endeavors, innovation could save lives and restore agency. The impact extended beyond the war. Wartime breakthroughs in industrial-scale production set the foundation for modern pharmaceuticals. Techniques developed to stabilize, ferment, and distribute penicillin became the blueprint for antibiotics in peacetime medicine. Millions benefited long after the conflict ended. The lessons of urgency, coordination, and ethical responsibility resonate even today, showing how science and industry, guided by human purpose, can overcome scarcity and crisis. Ultimately, the story of penicillin is a reflection on the fragility and resilience of life. War exposes humanity to its harshest extremes, yet it also illuminates the capacity for ingenuity and compassion. The men and women who labored to bring this medicine from lab benches to battlefield wards were not just scientists and workers. They were bearers of hope, rewriting the rules of survival. In 1943, America had no penicillin. By 1944, thousands of lives have been saved, limbs preserved, and morale restored. The ripple effects extended far beyond the front lines, shaping medicine, industry, and ethics for generations. In the intersection of science and war, of human need and technological mastery, penicillin became more than a drug. It became a symbol of what is possible when necessity, innovation, and humanity converge.